Good morning to one and all. This is Vaijayanti Patil from Turbe MPS. Welcome back to online education channel by BMS. Today we are going to deal with the third part of Introduction to Biotechnology, that is about plant tissues. After dealing with animal tissues, today we are going to start with plant tissues. We see plants also grow in size. They develop fruits, flowers. Today we'll look into details of how this takes place, or with help of which tissue does all this occur. Plant tissues are of two major types. First is the meristematic tissue. We'll look into details of it. First, we'll have an overview of this. Meristematic tissue is further divided into three types. One is apical meristem, second is the lateral meristem, and third is intercalary meristem. Second major type of plant tissue are permanent tissues, in which we have two subtypes. Simple tissue, which is further classified into other three types. One is Parenchyma, Collenchyma, and Sclerenchyma. The second subtype of permanent tissue is the complex tissue. In complex tissue, we have xylem and phloem. I hope students by now you have understood the difference between simple and complex tissue. Tissues which are made up of single type of cell are known as simple tissues, whereas the tissues which compose of more than two different types of cells are called as complex tissue. Now let us look into detail of all these different types of plant tissues. We have an activity in which two onions are kept in two different jars labeled as jar A and jar B. Jar A and jar B. Now, as we have to keep it in such a way that the bottom of the onion dips into the water. Now, due to the availability of water, the roots will start growing. We have to allow the roots to grow for 4 days, and after 4 days, we will cut the roots of the onion in jar B, as it is shown over here. Cut the root of onion in jar B. Again, we have to allow same thing, keep the onion back dipped into the water and allow it to grow for a few more days. While we have kept the onion for growth, we have to observe the growth of the root, that is length of the root. From this picture, it is clearly visible that slowly the roots grow. As we have cut the root tips of after 4 days, we have cut the onion or root tips of the onion from jar B. Even after keeping it in water for few days, we can observe that the root tips have not grown. Here, the root continued to grow, whereas after cutting the root tips, the onion in jar B does not show the growth. We can see it in the measurements. In jar A, the size goes on increasing, the length grows on increasing, whereas in jar B, there is no change. After cutting the root tips, it is same, 0.2, 0.2, 0.2. Now, why has this happened? Why has the root not grown of the onion in jar B? The reason behind that is the meristematic tissue. Now, let us understand what is meristematic tissue. It is a specialized tissue which can divide, redivide, and differentiate. The tissue which has ability to divide, redivide, or multiply and form new tissue or new cell are called meristematic tissue. There are some special characters of the cells in meristematic tissue. The cells of meristematic tissue contains very thick cytoplasm. It has conspicuous nucleus. Now see, this is the transverse section, a very thin section of the root tip. When we observe that section under microscope, this is how it looks. The dark purple color which we see in the center is the nucleus. The nucleus of meristematic tissue is very conspicuous. It can be easily seen. If you observe the cell wall of the meristematic cells are having thin wall. They are thin walled and are compactly arranged. You can see there is no space between these cells. Okay, so they are very compactly arranged. Vacuoles are absent, and these are highly active cells. Highly active for division, highly active for multiplication. Their number goes on increasing very rapidly. That's what we have seen in the case of the onion in jar 1. When the root tips were not cut, the root kept on growing continuously. Function of this meristematic tissue is to bring the growth of the plant. Again, in this meristematic tissue, there are 3 different types. First is the apical meristem. Apical, apex, meaning the tip of the stem or tip of the root is known as apical, and the meristematic tissues found in the tips of root or of the stem are known as apical meristem. Second is the intercalary meristem. Intercalary is the space at the nodal region where the leaves grow. Okay, so the meristematic tissue present at the node region is known as intercalary meristem. And the third one is lateral meristem. Lateral meristem is present on the lateral sides of the stem. We are also going to study each of these type in details and look out their functions. So as I said, first is the apical meristem, which is present either at the tip of the stem or at the tip of the root. So remember, location of apical meristem is always in the root tip or shoot tip. Function. Here you can observe that the shoot is coming out of the soil. On the same way, the roots are going deep in the soil. The roots are growing in length and going deep in the soil. So, function of apical meristem is to increase the length of the stem as well as length of the root. Apical meristem present in the stem helps in growing or increasing the length of stem. On the same side, the apical meristem present at the tip of the root helps in increasing the length of the roots. So it is the function of apical meristem. The second type is intercalary meristem. As I said, intercalary meristem is present at the nodal region where leaves grow. So, it is present at the base of petiole of leaf. Petiole meaning stalk of the leaf and of branches. Function of intercalary meristem is it helps in growth of the branches, formation of leaves and flowers and fruits. Okay, now see, we know that the stem grows from the nodal region. New leaves, they develop at the nodal region at the tip of the plant. Okay, so that growth of stem or formation of new stem is because of the intercalary meristem. If you observe in case of plants, the birds or the flowers, they usually grow either at the tip or at the axil, at the nodal region of the plant. Why? Because of presence of intercalary meristem. So, intercalary meristem is responsible for growth of branches and formation of leaves and flowers. The third type of meristem is lateral meristem. Lateral meaning the lateral sides. The meristem that is present on the lateral sides of the plant is known as lateral meristem. Function is to increase the girth, meaning diameter of the root and the stem. We know, see the big plants, the stem of the big plants are really very broad. For many plants, we can't even hold it in both of our hands. Okay, as the year passes, the diameter of the stem as well as the root, they go on increasing. This growth of diameter or this increase in the diameter is because of the lateral meristem. So it increases the girth of root and stem. It is very important for secondary growth in the plants. This is all about the meristematic tissue. Now, let us study the permanent tissues in the plant. Now, what is meaning of permanent? We have seen in meristematic tissue, the cells are capable of dividing or multiplying and forming new cells. They can keep on growing, they keep on multiplying. In case of permanent tissues, the cell loses its ability to multiply or to divide. So, permanent tissues, after their growth is complete, they cannot form new cells. They lose their ability to divide, but they start performing specific function and they occupy specific place in the plant. Thus, they permanently acquire a specific structure, shape, and location and perform a specific function. This is called as differentiation, and permanent tissues are formed from this differentiated cells. There are two types of permanent tissues. Simple permanent tissue and complex permanent tissue. Let us see the activity to study the simple permanent tissue. Now, student, this plant is the Rohio plant. If you observe this plant carefully, the upper surface of the leaf is green in color, whereas the lower surface of the leaf is purple in color. Okay. Hence, we generally use this plant to study the tissues of the plant. Now, what we have to do is take a fresh leaf of rohio, cut it in such a way, obliquely, such that we get a very thin section on the lower surface of the leaf. Place it on the slide and put dilute saffron in stain. Keep it in the stain for 1 minute. Let it stain. Spread it on the slide. Put a drop of glycerin and cover it with cover slip. The entire surface of plant is made up of only one layer. This is how it looks under microscope. When we take a very thin section on the slide, stain it and observe under the microscope, this is how it looks like. See the cell wall. Now the cell is hexagonal in shape. The cell wall is very thin. These are entirely filled with the cytoplasm. So it is having thick cytoplasm, thin wall, and very compactly arranged. You can observe there is no space in between the two cells. Here is one cell, other cell, third cell. There is very less or no space. So in the simple permanent tissue, these cells are compactly arranged and they are rich in cytoplasm. Now this tissue that we see over here is called as epidermis. Epidermis means the way we are having skin on our body. Similarly, plant is having a thin layer of single layered cell which is known as epidermis. This epidermis is also covered by a waxy layer which is known as cuticle. Now, cuticle helps in retention of water, okay, for maintaining the water content. See, now this is how the activity is performed. Hold the leaf or cut the leaf obliquely, take a thin section, put it in the saffronin, keep it for 1 minute, then take it again back into the water and spread it over the slide. Add a drop of glycerin and then cover it with the cover slip. After covering with the cover slip, observe it under the microscope. Epidermis is present on the entire plant from the stem to the roots. It is the outer layer of the plant. As I said our body is covered by skin. In case of plant, the plant's body is covered by epidermis. It is made up of one single layer of simple cells. Cells of epidermis are flat and have no intercellular spaces between them. They form single continuous layer. You must have observed during rainy season on many plants, the water droplets get arranged, or even if you go for a morning walk early in the morning in the winters, you can see such type of water droplets on many of the plants. Okay. Now, why is it happening so? Why that water is not getting absorbed within the plant or going off? It is because of the thin waxy layer. As I told you, here you can see the black color layer. This is a layer of wax which is known as cuticle, which helps in retention of water in the plant. It helps to save water loss in the plants. Now, let us see types of simple permanent tissues. In this, we have 3 types. Parenchyma tissue, which looks like this. Collenchyma, which look like this. And Sclerenchyma tissue, which will look like this. Remember, there are three types of simple permanent tissue: parenchyma, collenchyma, and sclerenchyma. Now let us see this in detail. First is the parenchyma tissue. Structure of this tissue is, they are called as living cells and they have thin wall. I would request you to observe the figures, the pictures very carefully. You have to notice the difference between the three tissues. Now here, this is the slide, the permanent slide of parenchyma tissue. When we observe under microscope, the parenchyma tissue will look like this. The cells are very compactly arranged. There is no space between the cells. Okay. They are rich in cytoplasm and they have very thin wall. If we talk about location of parenchyma, it is present in all the parts, including root, stem, leaves, flowers, and seeds. Function of parenchyma is to support storage of food and filling up vacant places within the plant. Okay, it provides support, storage of food, and filling the vacant spaces. Now there are again subtypes of parenchyma tissue. In parenchyma again we are having subtypes. First is Sclerenchyma. As I say, parenchyma cells are present everywhere in the plant. Okay. In this, Sclerenchyma is when the parenchyma is modified by the storage of chlorophyll pigment, because of which the leaves appear green. So, Sclerenchyma is the parenchyma with chlorophyll. It is present in the leaves. Chlorophyll is an important pigment required for photosynthesis. As leaves perform photosynthesis, that is the function of Sclerenchyma. The second subtype is Parenchyma. Here, the parenchyma tissue is modified for storage of air. Okay. The tissues have air space. The tissues have air spaces in it so that it can help the aquatic plant. Aquatic meaning the plants which are observed in water bodies. The plants that are present in rivers, ponds, or even oceans. In such plants, the parenchyma have air spaces, or the cells store air so that it can help the plant in floating by providing buoyancy. So, function of parenchyma is to help aquatic plants, leaves, and stems to float on the water or in the water. Now, in which plants do we see this? See, Sclerenchyma, so we observe in all the plants. We know that the leaves are green. This green color is because of Sclerenchyma. If we talk about Parenchyma, they are found only in the aquatic plants because it has to provide buoyancy to the plant for floating. So examples of Parenchyma are, this is the Ecornia plant, which you must have observed in the water bodies nearby, even nearby the railway track also, or in the lakes also. You can see this plant. These are water lilies. If you must have visited any temple, in temples, these lilies flowers are available. Okay. or in the lakes, lotus plant, वगैरह. If you open the stalk or the petiole of the flower, you can see the parenchyma. Now see, this is how the section of parenchyma look like. You can see it is like a sponge. It is very spongy because of the air spaces in it. Okay, so parenchyma is seen in the aquatic plants and it helps the plants in floating. The second type of simple permanent tissue is Sclerenchyma. Now this Sclerenchyma, they have slightly elongated cell. As you can observe it over here, the cell is slightly elongated. It is also a living cell and has thick wall. Now observe this wall. See, this is a section of Sclerenchyma. Compare with the parenchyma. I have shown you the slide of parenchyma and this is a slide of Sclerenchyma. This is how you can observe it under the microscope. This Sclerenchyma looks under the microscope. See, the wall is much thicker than the parenchyma. Okay, so it has thick cell wall and here you can see this red color structure. That is the deposition of cellulose and pectin. Due to deposition of cellulose and pectin, Sclerenchyma have thick cell wall. Location. Sclerenchyma are present at the base of leaf petiole, branches, and even stem. It provides support and flexibility. Support as well as flexibility to the various parts. Next is Sclerenchyma. Now, Sclerenchyma are called as dead cells. Notice that difference. Parenchyma and Sclerenchyma are the living cells, whereas Sclerenchyma are the dead cells. If we talk about structure, the cell is spindle shape. It is flat at the center and tapering at both the ends. This is called spindle shape fiber. Okay. In case of parenchyma, the cells were oval or pentagonal, hexagonal. In case of Sclerenchyma, the cells were elongated. And in case of Sclerenchyma, the cells are spindle shaped. Now, again observe the wall, cell wall of the Sclerenchyma. It is much thicker. Now, this is the section of Sclerenchyma observed under microscope. When we observe it under microscope, we see the cells appear brownish in color with thick wall. See, when the cytoplasm has dried up because it is a dead cell. The cytoplasm is entirely covered up by the pigment that is called lignin. It is a protein called lignin. Hence, they are called lignified cell wall. Now, if the simple example of this Sclerenchyma is, we observe the stems of the plants, they appear to be brown. Okay, the epidermis of the bigger plants, they appear to be worn out. That hard tissue of the stem that we see, it is the Sclerenchyma tissue, which we observe in the stem. Okay, not only stem, but it is also present in the veins of the leaves. It is present in the seed coat of any fruit, even the outer shell of the coconut. which is hard and brownish or the greenish in color. All this, whatever hard structure is observed in the plants, it is because of Sclerenchyma. Even jute is used for making bags and cloths. So that jute is also a Sclerenchyma fiber. Remember this. Function is it gives strength and rigidity to the parts of the plant. It gives strength and rigidity to the parts of the plants. This was all about simple permanent tissues. Now we are going to start with complex permanent tissues. In complex permanent tissues, as I said, as these tissues are formed of more than 2 different types of tissue, it is called complex permanent tissue. In this, we have 2 types. One is xylem and the other is phloem. Now, let us study both these tissues in detail. First is xylem. Now, it is characterized by thick wall, dead cells. Now, look at the cell types. Tracheids, vessels, xylem fiber or the dead cells, or it is also called as xylem sclerenchyma, and the fourth one is xylem parenchyma. So these are the 4 types of cell which makes the xylem tissue. Remember once more time, it is tracheids, vessels, xylem fibers or xylem sclerenchyma, xylem parenchyma or the living cells. Structure, it is interconnected tubes. You can observe it over here. Observe it over here. It is interconnected tubes. These are the tubes. They are connected to each other. These are the vessel cells. These are the vessels. Beside the vessels are the tracheids.
Thank you.
Okay, so this is the entire xylem. You can see the movement of the nutrients or the movement of the water in this. It always conducts the water or mineral nutrients in the upward direction. Remember this. Xylem conducts waters and minerals only in the upward direction, as you see over here. It is moving in the upward direction. Usually, it conducts waters and mineral nutrients. The second type is the phloem. It consists of cytoplasm containing living cell. This is how the phloem look like. Now there are again 4 components of the phloem. First is sieve tube and a companion cell. Now observe this figure carefully. This tube, you can see a plate between the tube. Okay, this is a tube. At both the ends you have a sieve plate having the pores or the holes on it. So they are called as sieve tube. The cell beside the tube is called as companion cell. Tube cell do not have its own nucleus. The companion cell nucleus serves to the tube cell, sieve tube. The third part or the third cell of phloem is phloem parenchyma, which helps in storage of nutrients. And the fourth type is phloem fibers, that is the phloem sclerenchyma or the dead cells. So these are the 4 cell types of the phloem: sieve tubes, companion cells, phloem parenchyma, and phloem fibers. If we talk about structure, tubes join to each other. Now here is the key. See. Sir, there is lots of disturbance. Please, can you mute the mic? Hello? Sir, please mute the mic. Now, these are the sieve tubes. These are the sieve plates. Now here you observe the flow of the nutrients. Now it is multi-directional flow. The nutrients will flow in all the directions, to the root as well as to the tip of the plant. Let us quickly recap what we have studied today. We have learned the simple, we have studied about the tissues in plants, in which first we studied about the simple or the meristematic tissue. Meristematic tissues are the tissues which can divide and redivide to form new cells and tissues. In this, we studied three types. First is the apical meristem that is present at the tip of the root and shoot. It helps in increase the length of root and stem. Second one, we have intercalary meristem, which is present at the base of the node, at the base of the petiole and leaf. It helps in growth of branches, formation of leaves and flowers. Whereas the third is lateral meristem, which is present at the lateral sides of the stem or the roots, and it helps in increase in breadth or diameter of the root and stem. Then we studied about the permanent tissues. Permanent tissues are the tissues that have lost its ability to divide. In this, we have two subtypes. Simple tissue. In simple tissue, again, three different types. First is parenchyma cell, which are oval or hexagonal in shape, thin wall and rich in cytoplasm. They are also called living cells, and there are of two different subtypes: collenchyma, chlorenchyma having chlorophyll, and erenchyma having storage of air, which is majorly observed in aquatic plant. This is how it looks like. The second one is collenchyma, which are again the living cells, but are thick-walled and elongated cells. These are usually found in the stems of the plant, and it provides support to the plant tissues. Third is sclerenchyma. They are also known as dead cell, no cytoplasm. They have very thick wall with deposition of lignin on the cell wall. The second subtype in the permanent tissue is complex tissue, which is formed up of more than two different types of cells. In this, we have studied two subtypes. First is the xylem. Xylem consists of xylem vessels. Then tracheids, xylem parenchyma and xylem sclerenchyma. Xylem helps in transportation of nutrients and water in upward direction, that is from the root to the upper parts of the plant. Whereas phloem is made up of sieve plate or the sieve tube, the companion cell, phloem parenchyma and sclerenchyma. Phloem is also conducting tissue. It helps in transportation of nutrients. The nutrients that are formed by the process of photosynthesis and respiration are transported from the upper part to the other parts. So here the transportation of the nutrient is in all directions. So it shows multi-directional flow. We have ended the assignment, students. What are the types of simple permanent tissue? Just now I have explained it. Just take a screenshot of this and complete this assignment in your book so that you can prepare for your exams.
नाही